All right.
And yeah, we heard from this was touched on a little bit by Sid and Zoe earlier this morning, and probably you saw their presentation.
Yes,
Um,
exactly.
so and I think Sarah is one of the folks who's doing all this work on sleep and KS, which, as we all know, is really important for patients and families. And so obviously you're going to go even deeper than Sid and Zoe did on the sleep work that you guys are doing. So I'll let you take it away.
Yes. So good afternoon, everyone. As Eric just said, my name is Sarah. I'm a postdoctoral fellow in the lab of Dr. Jonathan Lipton at Boston Children's Hospital. And broadly, the Lipton Lab is interested in understanding the interface between disorders of the developing brain and the circadian system. And so the work that I'm going to show you today, or share with you today, pardon, really offers a glimpse of our effort, along with our collaborators, Sid and Zoe and Joe at Boston Children's, to build a translational pipeline that allows us to study cells from patients in a dish all the way to clinical human populations. And so I'd like to start my talk off by saying that it has been really well established that sleep disturbances are highly comorbid with neurodevelopmental disorders and occur in approximately 50 to 90% of children with NDDs. Although this prevalence, the true prevalence, is difficult to ascertain. Nevertheless, its occurrence is in stark contrast to the 20 to 30% of typically developing children who also experience sleep disturbances. Now, these sleep disruptions are not without their consequences, right? They've been shown to exacerbate many of the core features of NDDs, increasing repetitive behavior, affecting daytime behavior, aggression, seizure threshold, and overall quality of life of the patient and their family members as well. However, research directly evaluating the circadian system in NDDs still remains extremely sparse. And if we take a step back for a moment, what we're actually asking is a much larger question. And that is, are sleep and circadian disturbances simply a byproduct of abnormal brain development, or do they actively contribute to disease pathogenesis? This is an immense but critical question to tackle. So when we refer to sleep, what we're actually talking about is the combined contribution of two systems. So that is a state-dependent sleep homeostat and a circadian oscillator. And these are known as process S and process C. So process S is basically a regulatory mechanism that is based on the prior duration of wakefulness. Essentially, the homeostatic drive to initiate sleep increases the longer you've been awake, and then it dissipates throughout the course of the sleep episode. However, process C, on the other hand, is largely independent of sleep history, and it represents the action of an endogenous biological pacemaker. So I think the easiest way to think about this is that the sleep homeostat tells us how much we need to sleep, but the circadian oscillator tells us when we need to sleep. And it's the when we need to sleep that I am focused on as a scientist. And so if we zoom in on process C a little more extensively, what we know is that the circadian system is a multi-scaled biological timekeeper that synchronizes homeostatic processes with the Earth's light dark cycle, thereby anticipating physiological need as a function of time of day. And I'm just going to see if I can bring up my pointer over here, pardon. Right. So the way this works is that photic information from the environment, so light information from the environment, is conveyed by specialized cells in the retina to a region of the mammalian brain known as the suprachiasmatic nucleus, or the SCN. Where here, the SCN's role is to generate, maintain, and synchronize rhythms across tissues and organs of the body to achieve a state of internal synchrony or internal harmony. And we know that stable circadian rhythms are really important for physiological and psychological well-being, but that alterations in these rhythms have been associated with several disease states, including various metabolic disorders, cancer, and of particular interest to our lab, neurodegenerative and neurodevelopmental disorders. So how does this really work at the cellular level? Well, in virtually all cells of the human body, there exists an evolutionarily conserved, self-sustaining, autoregulatory transcriptional translational feedback loop. And we call this loop the TTFL in the field. And this loop basically starts with the two transcription factors, BMAL1 and clock, that bind to the promoters rhythmically of thousands of downstream genes, including their own inhibitors, the PER and CRY gene families. And the subsequent protein products of the PER and CRY genes basically feed back onto this loop to suppress the transcriptional activity of BMAL1 and clock. But after some time, these PER and CRY complexes are ubiquitinated and they're degraded, which basically releases their inhibitory effect on BMAL1 clock, and it allows the cycle to begin anew. And this process takes approximately 24 hours to complete. So when developing a pipeline that would really allow us to dissect the mechanistic contribution of the circadian system to neurodevelopmental outcome, there was a strict set of criteria that we considered. The first is that we wanted to work with a neurodevelopmental disorder that had a strong sleep phenotype, and one in which the sleep phenotype was associated with neurodevelopmental trajectory. The third criteria is that we wanted to work with a neurodevelopmental disorder that had an identified cause and one in which the causative gene is.
Sarah?
Yes.
I'm sorry to interrupt. We have a request. So we have translators who are translating, and there was a request to speak a little slower if possible.
Oh, of course,
yeah.
not a problem.
Yeah,
sorry.
You couldn't have known. No worries. Just want to let you know. So if you can just go a little slower so the translators can keep up with you.
Absolutely,
Thanks.
no problem.
Okay.
So the fourth criteria is that we were interested in working with a disorder where that causative gene was ubiquitously expressed. And this is important because in order for the system to work with the model system we have in the lab, it was important that these genes be expressed everywhere in the body and not just in the brain or in neurons, for example. And I think this will make a little bit more sense when I describe what our model system is in the lab. And so you can imagine that this is an incredibly tough bill to fit, but this criteria, as well as our overarching question, was really strikingly exemplified in Kleischer syndrome. Now, I need not explain any of the clinical facets of Kleischer syndrome to anyone in this room, but suffice to say, what we thought was really interesting from a scientific perspective was this finding that during adolescence, a time of distinct changes to sleep and the circadian system, patients with Kleistra have been reported to exhibit an acute sleep crisis, which is then preceded by profound neurodevelopmental regression. And so really what Kleistra offers us is a window to understand sleep-related neuropsychiatric and neurodevelopmental compromise. Another way of saying this is how do sleep and circadian dysfunction contribute to neurodevelopmental outcome? And Kleistra syndrome really presented as the perfect opportunity to build this platform. So what we were able to do from there was we were able to basically collaborate with the Boston Children's Kleistra Clinic, which is the largest of its kind in the US. And as you just mentioned, you heard from Dr. Sid and Zoe just before. And what we did here is we leveraged the multi-scaled nature of the circadian system, which basically means that we can study rhythms of cells in a dish all the way to human populations by tracking biometric data. So by equipping patients with Fitbits, we're actually able to look at their rest activity cycles. And this really allowed us to build a multimodal translational pipeline, really. So what I mean by that is like really bench to bedside for understanding how the circadian system is mechanistically contributing towards a neurodevelopmental outcome. And so what Sid and Zoe are doing, as they touched upon, they're really responsible for the clinical phenotyping at the Boston Children's Kleistra Clinic. And then we've also collaborated with Dr. Joe Kozowski, who runs the DART lab, who's been able to actually analyze the patient biometric data that we've collected from all of the people who have willingly joined our study. And then over on the Lifton lab side of things, I've really led the cellular modeling and the phenotyping of both patient cells, but also understanding how the causative gene of Kleistra syndrome is involved in the regulation of circadian rhythms. And that's really the data that I'm going to be showing with you today. So the overall goal, I should say, before moving on, of this type of pipeline is really to be able to, I don't know, I'm zoomed in. I apologize. The goal of this type of pipeline is to really be able to cross-correlate the cellular phenotypes with the clinical phenotyping and the circadian biometrics of patients, with the goal of being able to stratify patients that are most at risk for developing the sleep-related neuropsychiatric compromise. And of course, the goal is to start with Kleefstra syndrome, but we hope that this uncovers generalizable findings that tell us something about how the circadian system actually contributes to neurodevelopmental disorders. And so I think a very logical question here, and where we really needed to start, was to ask the question, well, what role does EHMT1 play in the regulation of circadian rhythms, if at all? And to be entirely honest with everyone in this room, to date, there are no reported roles for the causative gene of Kleefstra, EHMT1, as everyone knows, in the regulation of circadian rhythmicity. But there are several convergent lines of evidence that do exist. The first is that we know that there is a very strong bidirectional relationship between histone modifications and circadian regulation. And this is important because we know that EHMT1 is an epigenetic regulator. The second is that there was a large-scale interactomic study that was made publicly available by another lab that basically indicated that EHMT1 and one of the core circadian proteins that I introduced before, clock, are believed to form protein-protein complexes in cells. And collectively, this data led us to our central hypothesis, which is a very simple one, honestly, and that's that disease-relevant mutations in EHMT1 impair circadian rhythms. But importantly, we further speculated that the loss of EHMT1-dependent circadian rhythmicity is a major contributor of post-pubertal onset of the sleep-associated neurological regression that we see in Kleefstra. Now, that's a mouthful. I'm definitely going to explain what that means as I go on. And so the very first experiment I did when I took on this project was to ask the question, as I just said, what role does EHMT1 play in the regulation of circadian rhythms? And the way I did this was I leveraged the kind of model system that we already have working in the Lipton lab. And so for those of us who study circadian rhythms in cells, the gold standard for working with these types of models is to use a cell line known as the U2OS cells. Now, I have to say that U2OS cells are an osteosarcoma cell line, but the reason that we use them is that they are incredibly rhythmic. They have really, really beautiful and really, really strong circadian rhythms. And what we have that makes these cells so special in the lab is that these cells have a circadian reporter system. So what they have is they have a PER2 luciferase system that is expressed in these cells. And basically what happens is that every time clock and BMAL1 bind to this PER2 reporter, we see a luminescent output. So essentially what this means is that we're able to look at cellular rhythms in real time. So I can plate these cells, I can synchronize them, and then I can put them in a luminometer. And this luminometer will measure how much luminescent output is coming from these cells. And then it essentially records this data where I can look at various circadian parameters of interest. So the very first thing I did with these cells is I tried to recapitulate the haploinsufficiency of EHMT1 that we see in Kleefstra patients. And the way I did this was by using siRNA to knock down the expression of the gene. And here, what you're looking at are just two such examples. I used two different constructs to knock down EHMT1. And this is the type of data that we get from the LumaCycle. So over here, what you're looking at in gray is you're looking at control cells that basically just received a scramble sequence. I'm not knocking down any gene in the control cells. But then here in pink and in purple, I'm knocking down EHMT1 with two different doses of that transcript. And what you can see is that we see a dose-dependent suppression of circadian amplitude. Now, I want to just take a moment to explain why this is important and what this means. So what we see is that the amplitude of the rhythm is less robust, right? So it's less strong. And changes to the amplitude of a circadian rhythm are typically ascribed to changes in the positive limb. So they're typically ascribed to the part that drives the rhythm, which is the clock BML1 portion of the loop. But what you'll notice is that we see no changes in the periodicity. We see no changes in the timing of the rhythm. And changes in the timing of the rhythm are often ascribed to changes in those per and cry complexes that feed back onto clock and BML1. And so, what this data preliminarily told us was that it seems that EHMT1 plays a role in the maintenance of normal circadian rhythmicity, but in particular, it seems to play a role somewhere in the positive limb of the TTFL, somewhere in the clock BML1 portion of this cycle, essentially. And so moving forward, we wanted to corroborate this data using another technique. And for some of us in the room, you might be familiar with a drug known as UNCO642, which is a selective inhibitor of EHMT1 and its partner, G9A. And I'm using this graph, which is published by another group, but I'm showing it simply to make the point that this drug is really selective for EHMT1 and G9A. So it doesn't seem to affect any of the other histone lysine methyltransferases, meaning that it has excellent selectivity. And we actually also know that it has low cell toxicity, making it an ideal drug for inhibiting EHMT1 in our cellular system. And so much like what I did with the siRNA is I took my U2OS cells, I plated them, and then I exposed them to UNC0642, I synchronized them, and I placed them back into that machine that monitors circadian rhythms. And what we found was that when we inhibited EHMT1, we found a dose-dependent suppression of circadian amplitude, but no changes in the periodicity, basically corroborating our initial findings. And a point that I just kind of want to make broadly over here is you'll notice that I had said that we exposed the cells, or rather we inhibited EHMT1 with UNC0642, and then we immediately placed them in the luminometer, and it immediately detected a decrease in amplitude. And that immediate effect is really important because while I was running these experiments, I conducted another experiment on the side to see how long I would have to expose the cells to UNC0642 for before we saw a global decrease in H3K9 Me2, right, which is the primary role of EHMT1. And what I saw is that it took a period of 4 days. And so the reason why this is so important to note is that the effect on the circadian oscillator that we saw when we inhibited EHMT1 was immediate, meaning that the role that it likely plays in the oscillator is independent from its role involving histone methylation, right? And so collectively, what this data essentially indicated to us, as I had said, is that EHMT1 seems to be involved in maintaining normal circadian rhythms. And in particular, it seems to be involved in modulating how strong the rhythm is. And this is really important because changes in the strength of the rhythm are often seen as rhythms that are less robust and less resilient, meaning that they are more vulnerable to perturbation. And this is something that we're going to talk about as I go through my data. So moving forward, now that we've established a role for EHMT1 in modulating circadian rhythms, I think the next question was to ask, even though EHMT1 seems to play a role in keeping rhythms in or I should say, keeping the strength of the circadian rhythm, it doesn't necessarily mean that EHMT1 interacts with BML1 or clock directly. So we wanted to test that. So to start with, we used super resolution microscopy and we used immunocytochemistry to look at the nucleus of cells where we used antibodies to label EHMT1 in green and clock in magenta. And any area where these two signals potentially overlap appear over here in white. And what you can do is you can draw a line through these overlapping signals, and you can measure the intensity of the magenta signal and measure the intensity of the green signal across that line. And then I can plot that data which is plotted over here. And what you can see is that they have the exact same profile, suggesting that these two proteins occupy the same space in the cell. And what that basically suggests, or the interpretation of this data, is that it's likely that these two proteins are interacting in the nucleus, right? So we love using super resolution microscopy. We love doing ICCs because they're really, really beautiful visualization tools. But something to keep in mind, and a potential limitation of these techniques, is that the nucleus is a really crowded environment. And so, although this is suggestive that these two proteins interact, it doesn't definitively tell us that the proteins are interacting, because it is possible that they're occupying the same space, but that they're not directly touching each other. And so we wanted to verify that this interaction actually happens biochemically. And so to do so, what we did is we harvested adult mouse brains. So we harvested these brains, we lysed them, and then we performed a technique known as immunoprecipitation. And what we essentially do in immunoprecipitation, and it's actually one of my very favorite techniques in the lab, is we're able to use an antibody to isolate EHMT1. And when you isolate EHMT1, what you can ask is, well, what other proteins are also associated with EHMT1? So here, what we did is we saw that if we isolated EHMT1 and we ran a Western blot, we could see a signal of the correct size. But then what we did is we also ran a Western blot to see if clock was also found to be isolated with EHMT1, and we found that was indeed the case. And for any other biochemists in the room looking at these blots, I have to say that I do know that the band here looks quite faint, but you'll have to believe me when I say that all of the circadian proteins are actually expressed at levels that are quite low in human cells and in brain. And so seeing a band of this sort, for anyone in my field, knows that this is really, really striking, honestly. So then we moved on to ask this question from the opposite angle. So we actually used cells that were donated to us by parents. So we used healthy skin cells. And this time we asked, well, if we isolate clock, will we see that EHMT1 is also there? And that was indeed what we found. So here we know that we isolated clock in the skin cells, and we were able to see a signal for EHMT1 at the right size. And so collectively, what this really tells us is that at endogenous levels, both in the brain and in human skin cells, we know that EHMT1 is interacting with a core component of the circadian clock, right? So we know now that EHMT1 is involved in regulating the clock, right? It regulates its amplitude, but it's also interacting with the core clock directly. And so this really allowed us to set up a really important experiment that allowed us to map the biochemistry of this interaction. And essentially what I mean by this is that this allowed us to ask, well, what parts of EHMT1 are important for its interaction with the core circadian proteins in the cell? And I know that this sounds particularly academic, but this is actually a really important question, because this, in a way, using a very blunt tool, recapitulates some of the patient mutations we see in the population. And understanding what parts of EHMT1 are important for its interaction with the circadian system might help us stratify patients that are most at risk for developing this neurodevelopmental compromise. And so what I did was I looked at the structure of the EHMT1 protein, and very bluntly, I categorized it into 3 regions. And so there's this very large N-terminal IDR region in EHMT1, which is then followed by a highly conserved anchorin repeats. And we know that anchorin repeats, just in biology in general, are really, really important for protein-protein interactions. And then finally, there's the C-terminal set domain, which we know is responsible for the dimethylation of lysine 9 on histone 3. And so from there, I basically kind of did a plug and play system where I removed certain parts from the protein. So here, this is what we consider the full-length protein. And then I have a variant where I've removed the anchor and repeats and the set domain. I have one where I've only removed the set domain, one where I've removed this large N-terminal IDR region, one in which I've removed the anchor and repeats. And then I have a control construct, which is just important for making sure that my circadian protein of interest, in this case clock, that its interaction is actually specific to EHMT1 and not any random protein that I'm overexpressing. And so I'm going to show a series of immunoprecipitation experiments, which to those of us who arguably aren't used to looking at black blobs all day, I'll kind of guide you through the important parts of this experiment. But essentially what I'm doing is I'm taking the different variants of EHMT1 that I've made, and I'm overexpressing them in cells, right? So here I have proof that I've expressed all of my variants of interest, and I've expressed them at the same time as I have overexpressed clock. And what I basically do is I isolate these EHMT1 variants, and then I ask the question, like I had done with the brain, if I isolate this variant, will I see that my protein of interest, in this case clock, will I see that it still interacts with EHMT1? And so what was really nice at first was I found that when I overexpressed full-length EHMT1, I saw that it was able to interact with full-length clock. So I basically corroborated what I had seen from the brain and from the parental skin cells. But then when I looked at the different variants of EHMT1 that I made, I basically found that if I removed the anchor and repeats and the set domain, I completely lost interaction with clock. And so it actually seems that clock can interact with either the set domain of EHMT1 or it can interact with the anchor and repeats. But if these two regions are completely lost, it can no longer interact with what's left of the protein. So moving forward, I did the same thing for clock's obligate partner, BMAL1. And I basically found that if I express full-length EHMT1 and full-length BMAL, what I found was that they also interacted, right? And so this is really important because now I know that EHMT1 interacts both with clock and BMAL1 in cells. But here, what I found was something really interesting. I found that what was happening was the exact opposite of clock. So here, if I removed this N-terminal IDR of EHMT1, I found that it no longer interacted, that BMAL no longer interacted with the EHMT1 protein. And I have a slide shortly that will kind of, that really paints a picture of what this might look like. And the last piece of immunoprecipitation data that I would like to show you is I did this for one final circadian protein, and this protein is known as NPAS2. And NPAS2 is a paralog of clock. So essentially what I mean is that if we completely knock the clock protein out of the cells, NPAS2 is able to replace it, so to speak. So it's able to interact with BMAL, and it can compensate for some of clock's functions. And essentially, I found that EHMT1 is also able to interact with NPAS2, and it seems that it binds to either the anchor and repeats or the set domain. So it behaves very, very similarly to clock. So that was kind of a nice finding. They corroborated each other. All right, but what does this mean, right? And so what we found now, again, to kind of re-summarize, is that EHMT1 plays a role in modulating the strength of the circadian oscillator, but it is also interacting directly with BMAL1 and clock. But it seems that it interacts with both proteins through completely different domains, right? And so this is kind of our working hypothesis. This is really the best way I can kind of depict how this might be happening. But in the nucleus, BMAL1 and clock are obligate partners, and they bind to the promoter sequences of DNA, right? Because their role is to promote transcription. And we think that EHMT1 kind of straddles this BMAL clock complex, where BMAL1 and clock are essentially interacting with different parts of the EHMT1 protein. And as I said, the reason why that this is so particularly important is that it really helps us identify patients who might be at risk. So to give you an example, a patient who might have a mutation in the IDR region, we would predict that it would completely lose its interaction with both BMAL1 and clock. And so now we're really able to ask the question, well, are these patients most at risk for developing sleep-related neuropsychiatric compromise? And so we're now at the point working with Sid and Joe that based on this biochemical mapping, we can actually go back and look at the rest activity cycles of those patients and see if these patients have worse circadian rhythms than other patients that perhaps only have mutations in the set domain, right? That's just really to kind of give you an example of why this data is so particularly important. And so for today, I'd like to finish on two last experiments that also really help us conceptualize why this information is so particularly important in helping us build a translational pipeline. And so in the final set of experiments I would like to show you, we ask the question, all right, so we know that EHMT1 and circadian rhythms interact, but how do they interact to modulate downstream pathways? And so what we did is we leveraged data that was published by a colleague of ours that basically reported that the EHMT1 paralog in Drosophila is involved in modulating inflammation. So what they had basically found in Drosophila was that EHMT1 is involved in the regulation of stress-specific pathways. And so what we did was a very simple experiment. So we took our U2OS cells and we either collected them at baseline conditions, which is to say at 37 degrees in their incubators, or we exposed these cells for 1 hour to an oxidative stressor. So in this case, we use sodium arsenite. And what you can see from baseline to stress conditions is that there's a massive increase or a massive potentiation in EHMT1. And so we then asked the question, well, how does clock modulate EHMT1's potentiation? And so what we essentially did is we used genetic tools to knock the clock out in these cells. So in this case, we knocked out BMAL1, and we saw that this effect was completely abrogated, suggesting that EHMT1's response to stress is dependent on the presence of a functional circadian oscillator. And I think I'd just like to make a note that I think a really logical question here would be, well, how do we know that it's not about BMAL1 directly? We also have shown that in clock knockout cells. So if we knock out clock instead of BMAL1, we get the exact same phenotype. So it's really about abolishing a circadian rhythm in these cells that prevents EHMT1 from being able to mount a normal stress response. And so this led us to our working hypothesis, which we basically we're working on now to look at how this might be working in patient cells and in Kleefstra broadly. But what we think is happening is somewhat of a two-hit hypothesis, where we know that EHMT1 is important for the strength of a circadian oscillator, but we also know that the oscillator is important to help EHMT1 mount a stress response. So we think that what might be happening in Kleefstra syndrome is that there's haploinsufficiency of EHMT1. We have less EHMT1 to begin with. And so this means that patients have a less resilient and less robust internal oscillator, which basically makes them vulnerable to stressors. And then at some point in their life, there is some type of stressor. It could be an acute illness. It could be even just the hormonal changes that occur with adolescence itself. And since now we know that a normal circadian rhythm is required to mount this EHMT1 response, since they have a dampened rhythm to begin with, this response, we don't see this response from EHMT1, meaning that they have an impaired response to the stress pathway. A much simpler way of saying this is that because they have a dampened circadian rhythm, they're more vulnerable to stressors and they can't help EHMT1 mount the homeostatic response it would need to do in order to cope with this external stressor. And so the final experiment that I would like to show you today is, I think, a really good question as well. How does this work in the patient cells that we have in the lab? And so what we did is we took some healthy parental controls and we did the exact same experiment with them. We either expose these cells to baseline conditions or we expose them to an oxidative stressor. And you can see that even in healthy parental cells, there's a potentiation, there's an increase in EHMT1 in response to that cellular stressor. But now what happens in the patient phenotype, in the patient cells, pardon. So remarkably, what we saw is that this effect was almost completely abrogated in the patients. And even more interestingly, we saw that this seems to be genotype specific. So here what you can see is that patient 2 and patient 10 don't really, their baseline and their stress conditions don't seem to be very different from each other, whereas patient 1 is able to somewhat mount a response. And it turns out that patient 2 and patient 10 are individuals who have a mutation within that N-terminal IDR region. So again, in understanding how EHMT1 and the circadian system interact, this also helps us understand what patients might be at risk for this neurodevelopmental and neuropsychiatric compromise. And so we've really spent the last year and a half really trying to understand what role EHMT1 plays in circadian rhythms. And I think we're at a point to really test how those rhythms are changed in patients and what does this mean for their future neurodevelopmental trajectory. And so with that, I would just really like to acknowledge a few people. First and foremost, of course, the Lipton Lab and my supervisor, Jonathan, who has provided me with an incredible opportunity to work on a pipeline that virtually doesn't exist in the circadian field. And of course, our collaborators, Sid and Joe, who are experts and incredible in their own fields and have been amazing collaborators. I'd also like to thank our funding sources, and in particular, the Tommy Fuss Foundation that supports me as a postdoctoral fellow in doing this kind of work. And most importantly, I would like to acknowledge all of the parents and the patients who have donated their biometric data and their cells to this pipeline. I want you to know first and foremost, that this has been an invaluable resource, and we're working really, really hard to continue to build this pipeline, which thus far has been quite successful. So with that, thank you so much, and I'm happy to answer any questions.
Wow, Sarah, this was amazing. You completely blew my mind.
I'm glad it resonated.
And I think it's such a good example of kind of, we were talking earlier in the day, I was talking about kind of the halo effect of having our clinic at Boston Children's, and it was really cool how that's you and Jonathan got interested in Cleastra because the clinic was there and the patients were there, and then you guys started working on this, and now you've made these really exciting discoveries. And yeah, I have a few questions. I haven't had any questions come up on the chat, but I was wondering fundamentally, when you're doing these circadian experiments with cell lines, how do you sync the cell lines so they're in the right circadian rhythm? Because they don't have eyes, right? So they can't see the light and know when they're supposed to be asleep and awake.
Yeah, so that's a great question. There's multiple ways that we can synchronize cells. What I typically do is I use dexamethasone, which is actually a glucocorticoid. And what that basically does is capitalize on what we know about how peripheral organs of the body are synchronized. And what I mean by that is that the SCN, right, in our brain is synchronized by light, but there are other organs of the body that don't receive this light information. And so the SCN needs to synchronize them in another way. And what we know is that in the morning, we have this rise of cortisol, and then in the evening, we have this trough of cortisol. Well, actually, what it does is, I'm going to use the adrenal glands, for example, is that it's actually able to bind to glucocorticoid receptors. And what it does in binding to glucocorticoid receptors is it actually synchronizes that BMAL1 and clock cycle. And so we've basically found that U2OS cells also have those receptors. And so we can use a synthetic glucocorticoid to get the same effect.
Yep. So they do see the light. They just don't really directly see the light.
Exactly. Exactly. That's a great way of saying.
Yeah. Okay, cool. I had another thought, which was, is there any possibility that EHMT1 is actually methylating clock? Because it is a methyltransferase, and we know that it can be promiscuous, and it does methylate proteins as well. Yes. Other than histones, obviously, I should say. I mean, histones are proteins, but other proteins have been shown to be methylated. So,
yeah. Like you said, it's known to do that. And it's also known to do that, particularly for transcription factors, for which clock is
Mhm.
one of them. So we've asked this question before, and I think it's just the techniques to do this right now are just a little bit, what I would say is they're a little bit dirty. And so the results that we got are a little bit ambiguous. So it's
Mm.
still a little bit hard for us to say whether EHMT1 is directly methylating BMAL1 and clock, but it is something we are interested in figuring out. And there are some ways that we can do that, but we don't know right now.
Yeah, it's really interesting. It raises so many exciting new questions. So there's a question that came up in the chat. I'll just read it exactly.
Mhm.
Since UNC specifically works by preventing H3K ME2, H3K9 ME2 deposition, why do you think the circadian effect is independent of K9 ME2? Is it possible that there isn't a global effect, but locally some genes lose K9 ME2 rapidly?
Yeah, that's a really, really, really good point. And I will say that we can't disambiguate at this point in time. And I think the next set of experiments for us is really to do transcriptomics. And one thing that we floated is instead of having an EHMT1 knockout cell line, we could potentially just treat cells with UNC0642 and ask that exact question. How does acute administration of UNC0642 locally change methylation, and is that mechanism that is behind like what is changing the circadian amplitude? So I will say that that's an excellent question to whoever put it in the chat. The answer is we don't know, but at least we know that it's not global rhythm. It's not global methylation, but it certainly might be local. That is a good point.
Interesting. Other thought that I wanted to ask about is we've been talking about biomarkers, and presumably this mark is showing up. This effect, I should say, is showing up not just in wouldn't just be in neurons because you're using
Yeah.
a model based on an immortalized cell line as well. Could
Mhm.
that become some kind of a biomarker where if we had an agent that would restore HMT1 function to the normal level, could you say, well, we are seeing that we are restoring circadian rhythm, and that is something that we know is problematic. And so therefore,
Yeah.
this is a biomarker we could think about validating in the future. It's a thought, a shot in the dark, but um,
I think it's a really good question. And I think that's been something that we've been starting to work with the patient lines, right? Because so many individuals have donated their cells. We
mhm,
can actually stably express our circadian reporter in those cells. So in the cells of individuals, I can actually look at their circadian clock in real time. I think the only the hiccup that we're currently dealing with in order to be able to, you can imagine someone could come into the clinic, donate some of their skin cells, and then in the lab we would be able to see if it's a biomarker by being able to tell whether their own circadian rhythm is altered in some way. I
mhm.
think at the moment the only place that we're really working hard to kind of overcome this limitation in the lab is in order to trial these types of studies, the best control would be an isogenic line. And so we're currently working with some other teams at Boston Children's to see if we can correct the mutation in the patient line to see how this would actually change their rhythm. So it's really a question of controls, but it certainly could be a very strong biomarker in the future.
When you get fibroblasts from the patients who have donated them, are they initially already on their circadian cycle and you can capture that as you initially grow them, or do you still have to synchronize them?
So
Um,
we still have to synchronize them.
okay.
Still to synchronize them. However, there are some other groups just in the circadian field that do exactly that. They have patients come in at an exact time of day. For example, they might collect saliva or they might collect blood, and they're actually able to look at what circadian time it is of that an individual person. And there's some
Yeah,
really big groups that are actually working on that, and so far they've been quite successful.
fantastic.
Yeah.
Yeah, yet another moonlighting role for EHMT1. I think you know that it's also been shown to interact with NF-kappa B, the
Yes.
NF-kappa B pathway, and there's
Yes.
ongoing work to try to refine that understanding further. So we keep seeing that it just does all these things, and it is an ancient gene. I was looking up how far back. It's basically metazoans. So it's
Yes.
like there's an EHMT, something that looks like an EHMT. Well, obviously, there's a one and a two in humans because it's duplicated. But and so it looks like as long as there have been complex multicellular animals that had to deal with things like day-night cycle. There has been an EHMT sitting around helping them do that. So it's remarkable how connected it is with so many functions.
Exactly. And I think what's so particularly interesting as well is that not only does EHMT1 seem to, as I said, be involved in regulating the circadian system, we know, as you're referring to, that EHMT1 is also involved in the regulation of stress-specific pathways, which we know that these stress-specific pathways are also under strong control of the circadian system. And so it becomes
Yeah,
kind of like this input versus output cycle that you can imagine, when corrupted at any point, can lead to like a myriad of downstream situations that are not homeostatic, right? So
yeah.
essentially an impaired response. And so this really becomes a wealth of information for us to dig through, but that we fundamentally think will be really, really important for patients.